The insect body has a hard outer covering called the exoskeleton. The exoskeleton is made of both chitin and protein. The exoskeleton acts like both a skin and a skeleton. It gives the insect form like a skeleton, but like a skin, it protects against water loss, injury, and infection by microorganisms. The insect body is divided into three major sections: the head, the thorax, and the abdomen. Each of these sections is further divided into segments. Insects have 6 legs. One pair of legs is attached to each of the 3 segments that form the thorax. Only adult insects have wings. Flies have 1 pair of wings attached to the thorax. All other insects have 2 pairs of wings attached to the thorax. The antennae are olfactory organs for detecting odors. Compound eyes are the main organs for vision. Spiracles are openings in the side of the insect that allow air to enter into the respiratory system. Let us examine the internal structure and the function of the organs in the insect body. Our study of insect internal structure and function will start with the respiratory system. The role of animal respiratory systems is to carry oxygen to the body's cells for oxidative metabolism. In insects, spiracles open to the atmosphere and attach to a network of silvery white tubes called trachea. Air containing oxygen enters the spiracles, and the oxygen diffuses along the tracheal tubes to the cells. Unlike vertebrate animals, insect blood does not have hemoglobin and does not carry oxygen. Since fine trachea extend to all the body cells, they also serve as a connective tissue that holds the internal tissues together. The fat body is the major tissue for insect metabolism and the storage of excess nutrients. Most of the fat body is in the abdomen surrounding the gut, where it can quickly take up and metabolize absorbed nutrients. In addition, fat body is also scattered throughout the body near other tissues that use its products. The metabolic function of the fat body is similar to that of the vertebrate liver. Like the liver, it stores excess nutrients and it synthesizes proteins, lipids, and carbohydrates that circulate in the insect blood for use by the other tissues. The insect nervous system conveys and integrates information about the internal and external environments and determines insect behavior. The central nervous system consists of the brain and the ventral nerve cord. Here is a detailed top view of the central nervous system. The brain consists of dumbbell-shaped visual lobes with nerves that project outward to the retina of the compound eyes. The ventral nerve cord consists of a series of ganglia, usually 1 per segment. Ganglia are thickened regions of the nervous system that contain the cell bodies of the nerve cells. The ganglia of the ventral nerve cord are connected by paired nerves called interganglionic connectives. Finally, the brain is connected to the ventral nerve cord by a pair of nerves that pass around the esophagus and connect to the subesophageal ganglion. Like other animals, insects possess an array of hormones that regulate their diverse physiological and biochemical processes. Hormonal sources in insects include the neuroendocrine system, the corpora lata, the prothoracic glands, and epitracheal glands. Other endocrine cells are also found in the gut and ovaries. The neuroendocrine system consists of nerve cells that secrete hormones. Neurosecretory cells are located mainly in the brain, the ventral nerve cord, and the corpora cardiaca. They are also found in association with other nervous tissues located throughout the body. Neurohormones are the master regulators and control most physiological and metabolic processes, including regulating secretion of the hormones that control molting, metamorphosis, and reproduction. Neurohormones also regulate the synthesis of blood lipids, carbohydrates, and proteins, and control energy metabolism related to flight. And they also control other basic physiological functions such as feeding activity and excretion. Corpora cardiaca are major neuroendocrine structures attached to the brain. Neurosecretory cells located in the brain synthesize and transport neurohormones to the corpora cardiaca, from which the brain hormones are stored and released. In addition, corpora cardiaca contain intrinsic neurosecretory cells that also synthesize and release neurohormones. The corpora lata are structurally associated near the corpora cardiaca, but they are not part of the neuroendocrine system. Corpora lata synthesize and secrete juvenile hormone. Juvenile hormone prevents immature insects from undergoing metamorphosis into premature adults during molting. Juvenile hormone also stimulates egg formation in most adult female insects. The prothoracic glands are a grape-like cluster of cells surrounding the trachea in the first thoracic segment. These glands secrete ecdysone, a hormone that stimulates the molting events necessary for insect growth. Prothoracic glands deteriorate in adult insects because adult insects no longer molt. Like the prothoracic glands, epitracheal glands are groups of secretory cells associated with the trachea. They secrete hormones that regulate molting behavior. Endocrine cells are also found in the gut and may affect feeding activity. The roles of the circulatory system are to transport essential metabolites from the fat body to the cells, carry waste to the excretory system, and provide immunity to harmful organisms. Insects have a simple, open circulatory system. The circulatory system consists of a dorsal vessel running the length of the body. The dorsal vessel is divided into a posterior heart that contains intake valves called ostia and an anterior aorta. The open space of the body is called the hemocoel. The hemocoel is filled with insect blood called hemolymph. Since insect hemolymph does not transport oxygen, it does not contain hemoglobin, and therefore lacks the red color that is characteristic of blood from vertebrate animals. Hemolymph is pumped forward by the heart through the aorta into the head and flows back through the body in the open hemocoel. Hemolymph re-enters the posterior heart through the ostial valves, and the cycle repeats. The hemocoel is always full of hemolymph, and the heart ensures its mixing. Auxiliary pulsatile hearts at the base of the antennae, legs, and wings pump hemolymph into those appendages. Although all insects have common structures in their digestive systems, these structures can be highly variable between species based on the diversity of foods that insects eat. We will use the grasshopper to illustrate the basic principle for these common structures. The insect digestive system is divided into three main sections: the foregut, the midgut, and the hindgut. The foregut is comprised of the mouth for ingesting food, the pharynx and esophagus for transporting food, and the crop where food is stored prior to digestion and absorption. If the food is solid, it moves from the crop into a muscular proventriculus, where it may be further ground before passing through the stomatidal valve into the midgut. The midgut is the region for the digestion of food and the absorption of nutrients. Finger-like projections, called gastric ceca, occur at the junction of the midgut and proventriculus and serve as major structures for nutrient absorption. Unabsorbed food particles pass through the proctodeal valve into the hindgut. At the junction of the midgut and hindgut are a group of long, thread-like tubules that flex about in the hemolymph. These tubules are called Malpighian tubules, and they are the insect excretory system. Malpighian tubules filter the hemolymph and form a urine containing water, salts, small metabolites, the nitrogenous waste products, and any toxic chemicals that are present. Undigested food from the midgut along with the Malpighian tubule urine are passed into the hindgut. Special cells in the rectum selectively reabsorb the water, salts, and small metabolites, and the remaining waste products are excreted. By selectively taking all water, salts, and small molecules into the Malpighian tubules, then selectively reabsorbing the water, salts, and metabolites in the hindgut, the insect can rid its body of waste and any unanticipated toxic chemicals while maintaining a normal metabolite and water balance. Most insects reproduce sexually and lay many eggs. The female reproductive system consists of paired ovaries made up of separate tubules called ovarioles. Ovarioles are divided into chambers called follicles. Each follicle contains an oocyte that is becoming mature by depositing yolk. Mature oocytes are present in the basal follicle. Mature oocytes are chorionated in the follicle, then passed into the lateral oviducts and to the common oviduct. Sperm are released from the spermatheca to fertilize the egg as it passes through the common oviduct for oviposition. Accessory gland secretions assist egg laying. These products may be venoms, as in the case of wasps, or cement to fix the egg to the oviposition site. In males, testes also consist of follicles where the sperm are matured as they progress from the tip to the base of the follicle. Mature sperm pass from the follicle to the vas deferens into the seminal vesicle. Male accessory glands produce products that mix with the sperm to protect and preserve the sperm. Some insect species produce a spermatophore that encloses the sperm and is passed to the female during mating. Other insects transfer sperm without a spermatophore. Accessory gland secretions may prevent the mated female from mating again by forming a temporary plug or by transferring chemicals that suppress mating behavior. Finally, the sperm or spermatophore are passed through the ejaculatory duct during copulation. Most insect species have wings as adults and are able to fly. Unlike birds and bats, insect wings are not modified forelimbs, but are extensions of the cuticle of the meso and metathoracic segments. These two thoracic segments also have prominent muscles used for generating the wing beat. Flight muscles of bats and birds attach directly to the wings and pull the wings up and down. In insects, only dragonflies and damselflies have muscles attached directly to the wing, and these muscles only produce the downstroke for the wing beat. In all other flying insects, both the downstroke and upstroke of the wing beat are produced in response to contractions by muscles that attach to the thoracic cuticle and not directly to the wing. The downstroke is produced by a set of dorsal longitudinal muscles attached to phragma. Phragma are cuticular invaginations of the meso and metathoracic segments. The upstroke is generated by a pair of dorsoventral muscles attached to the top and bottom surfaces of the meso and metathoracic segments. These indirect muscles act by undergoing rapid antagonistic changes in tensions that produce alternating changes in the length and height of the thoracic segments. These alternating changes in the shapes of the segments cause the base of the wing to move in and out over a lateral fulcrum point that flips the wing into the upstroke and the downstroke for the wing beat. Many insect species are able to move their wings rapidly and can fly at wing beats of 100 to 700 per second. By comparison, hummingbirds fly at approximately 50 wing beats per second. Muscles that attach directly to the base of the wing cause wing folding or may control the pitch and twisting of the wing in some species.